The CRC has given us the opportunity to bring together people from the different sectors, the industry, government, university, to focus on the challenges presented by a number of global trends in climate change, in trade in particular, where we're going to see the constant movement of organisms that were once confined to certain geographic areas. We are dispersing them around the planet. And we know from a lot of experience that it's extremely important to find these things quickly so that we can manage them when they're small, rather than after they begin to spread and the problem becomes more challenging to contain them. And basically there's a transition period where if it gets too large a problem, you can no longer eradicate. So what we want to do is develop the technologies and the tools that allow us to find these things quickly and accurately identify them, because you need to know what you're working with in order to respond appropriately. So we've been using some newer technologies called next generation sequencing, but also using them in certain ways to identify the best region of a genome to base a diagnostic on, so that we minimize the false negatives and the false positives that are common with diagnostic tools. The false positives are a problem because you end up incurring costs from unnecessary mitigation measures. You perhaps stop trade. The false negatives are a real problem because you end up with an incursion, and if not dealt with quickly, you're going to have high costs from impacts. So the tools we're developing are based on a new way to do polymerase chain reaction. You use enzymes that don't require the cycling. There are a lot of advantages to that I won't describe, but a lot of advantages to that. And there are some newer technologies that allow us to move those from the laboratory to the field. Again, the benefit of that is that we're getting highly accurate, very quick results in the field as to the identity of the organism we're dealing with. So we've been targeting an array of pathogens. We wanted to see if these technologies can be applied to several different types of organisms. So we're looking specifically at pathogenic bacteria. So we've selected targets that are gram-negative, gram-positive, that are vectored by arthropods, that are vectored by nematodes. We selected targets that are non-culturable, which present some of the biggest challenges in diagnostics. So we've looked at examples or representatives of each of these types of diagnostic targets, and we've applied these tools, and the progress has been staggering in the last 2 years. We've assembled a team of very young scientists that have done some amazing research. And with that research, we have now started to engage the end user communities, the people who do biosecurity surveillance, the people who are responsible for ports of entry. We're starting to engage them in how to use these tools effectively in their respective environments. And the results have been very positive. These tools are very robust. We can take them to the field and they perform as well as they do in the laboratory, again giving us very quick results, but most importantly, very accurate results. These technologies are really quite simple. We set up a display here to demonstrate these technologies. Everybody can take a crack at it, but they're very simple to use. 5 steps is all it takes to process your sample, and in a very short period of time you get your answer. So the benefit of that is twofold. Not only do you not want to import a pest that can cause a lot of damage, you don't want to export one and ruin your reputation and your marketing plan. So yes, the farmers will be able to use them, consultants will be able to use them, people who do field surveillance. It's a technology that's very portable. I have a device that I carry in a small case, goes in the overhead bin in an airplane. I travel with it with ease. These are very portable technologies. I think the ultimate goal then is to prevent shipping things out of a country. So our inspections on the point of embarkation is probably the most important, so that we're not pushing things out of areas without knowing it. Because The food distribution system, the plant distribution system is so sophisticated that we move plants and plant products from one continent to another in 48 to 72 hours, which is much shorter than the period necessary for it to express symptoms. So oftentimes we're moving plants around that are infected, but they're not showing symptoms, so we don't pull them out. These types of tests will allow us to detect pathogens in plants pre-symptomatic, and that's part of the power of it. Some of the real benefit of the CRC system is providing the platform that allows all these different perspectives to come together and develop a common plan. How are we going to tackle this problem? How are we going to implement the result? We're pretty good in academia with generating solutions that nobody ever uses. And so the mission of the CRC is to make sure that it doesn't sit on a shelf, that it gets out. And I think that's one of the great benefits. The second great benefit is we've seen a huge decline in expertise globally in certain disciplines. Bacteriology is one. These two projects, PBCRC 2002 and 2156, have allowed us to train and mentor 9 scientists that will be this generation's plant bacteriologists, restoring an expertise that was on the decline. I think the CRC should be very proud of that.